Bismillahirrahmanirrahim. I am Aisha from Department Chemistry, and my topic of presentation is mass spectrometry. In this presentation, we will discuss about the introduction of mass spectrometry, that what is mass spectrometry, then we will discuss about its principle, then instrumentations, how its parts are assembled, and at the last we see about its costing. First, we see the definition of mass spectrometry. So what is mass spectrometry? Mass spectrometry is an analytical technique used to determine the mass and composition of molecules and atoms. It involves the ionization of samples followed by the separation and detection of ions based on their mass to charge ratio. The process of mass spectrometry involves several steps, in which first of them is ionization. The sample is vaporized and ionized to create charged particles. This can be achieved through various ionization methods such as electron ionization, chemical ionization, electrospray ionization, or matrix assisted laser ionization. Second one is accelerated. After ionized, the ions are then accelerated in an electrical magnetic field, causing them to separate based on their mass to charge ratio. Heavier ions are deflected less, and lighter ions are deflected leading to their separation. Then detection. The separated ions are detected by a detector, which generates an electrical signal proportional to their abundance. The common detectors we used include electron multipliers, photomultiply tubes, and ion counters, which we discuss later. Then data is analyzed on the computer. Electrical signals are converted into mass spectra, which represents the abundance of ions and different mass of charge values. These spectra can be analyzed to determine the elemental composition, molecular weight, and structural information of the sample. This is the basic overview of mass spectrometry. Now we see why we use mass spectrometry. So there are several other spectroscopy techniques like NMR, IR, and UV visible spectroscopy. So why we use mass spectrometry? There are several reasons of using mass spectrometry. First of them is molecular identification. Mass spectrometry provides precise molecular identification by measuring the mass to charge ratio of ions. This is particularly useful when dealing with complex mixture or unknown compounds where other spectroscopy techniques may not suffer the same level of specificity. Second one is high sensitivity. Mass spectrometry offers exceptional sensitivity, allowing for the detection and quantification of trace amount of analytes. This is particularly important in fields such as pharmaceutical analysis, environmental monitoring, and metabolomics, where lower concentration of target compounds needed to be measured. Third one is structural elucidation. Mass spectrometry is very helpful in structural elucidation through fragmentation patterns obtained through ionization. This allows for the elucidation of chemical structures and the identification of functional groups and their connectivity within molecules. In contrast, other spectroscopy techniques may provide more limited structural insights. Then a wide range of analytes can be detected with the help of mass spectrometry. Because it is highly versatile and can analyze a wide range of analytes, including small molecules, large biomolecules such as proteins, peptides, nucleic acid, inorganic ions, and complex mixture. This broad applicability makes it a valuable tool in various fields such as pharmaceuticals, proteomics, metabolomics, environmental sciences, and forensic analysis. Then mass spectrometry is well suited for isotopic analysis. It can determine the isotopic abundance of elements within a sample. This is particularly useful in geochemistry, environmental science, and forensics, where isotopic ratio can provide valuable information about the origin, source, and processes involved in the formation of compounds. Then quantitative analysis. Mass spectrometry can be used for accurate and precise quantitative analysis for allowing for the determination of compounds concentration in a sample. Isotope dilution and stable isotopes labeling techniques can enhance the accuracy of quantitation. Other spectroscopy techniques such as UV visible spectroscopy or fluorescence spectroscopy may not offer the same level of accuracy or sensitivity for quantitative analysis. So that's why we use mass spectrometry. Then we discuss about the instrumentation of mass spectrometry, in which first of them is sample introduction system. This system includes devices for introducing the sample into the mass spectrometer. It can include syringes, sample loops, gas chromatography columns, liquid chromatography columns, direct infusion system, or solid sample probes. Second one is ion source. The ion source is responsible for converting the sample into ions. Different ionization techniques are employed, such as electron ionization, electrospray ionization, chemical ionization, atmospheric pressure, chemical ionization, matrix assisted laser desorption ionization, etc. These are further several types of ionization source: electron impact ionization source, electrospray ionization source, atmospheric pressure chemical ionization source. In first of them, electron impact ionization source. In this technique, high energy electrons bombarded the sample, causing ionization. Companies like Thermo Fisher Scientific and Agilent Technologies manufacture this ionization source. Then electrospray ionization source. ESI generates ions by spraying a liquid of sample through a charged needle, creating a fine aerosol of charged droplets. The companies which manufacture them, such as SCIEX, Thermo Fisher Scientific, and Agilent Technologies, produce ESI sources. Then atmospheric pressure chemical ionization source. This ionization occurs at atmospheric pressure, where a vaporized sample interacts with ions formed by a corona discharge. Companies like SCIEX, Thermo Fisher Scientific, and Agilent Technologies offer this ionization source. Then photoionization. In photoionization, the sample is irradiated with photons of scientific energies, causing the ejection of an electron and the formation of ions. This technique is often used in specialized applications such as photoionization, mass spectrometry, or laser desorption ionization. What is its function? The ion source is a crucial part of mass spectrometry that facilitates the conversion of neutral sample molecules into ions. It plays a key role in determining the ionization method employed and can significantly impact the sensitivity, selectivity, and efficiency of the analysis. This is the ion generation diagram, how ions are generated or ionized in mass spectrometry. And in this ion generation, we use 70 volts. So why we use 70 volts? Electrons with an energy of around 70 volts have enough energy to ionize most organic and inorganic molecules. At this energy, electrons can effectively remove an electron from the molecules, resulting in the formation of positively charged molecular ion. It also promotes fragmentation. The energy of the electrons in source is also efficient to induce fragmentation of the molecular ion. This fragmentation can provide valuable structural information about the analyte. The fragmentation pattern obtained in AI can aid in compound identification and elucidation of molecular structure. This is a diagram of structure 3,4-dimethoxy acetophenone. In this diagram, this is clearly shows the fragment how it is ionized, and by the process of mass spectrometry we can know the structural elucidation of this molecule. Though, which are the companies which manufactures this source? First of them is Extrail CMS. Extrail CMS is a company that specialized in mass spectrometry instruments and accessories. They offer a range of AI sources suitable for various gas chromatography mass spectrometry system. Then second one is IonSense. IonSense is a company that focuses on direct analysis in real time and other ionization techniques. Third one is V&F and Mesting GmbH. This company is a German-based company that specializes in manufacturing mass spectrometry ion sources, including EI sources. Then Gaia Science PT Ltd company. This company is also supplier of scientific instruments and laboratory equipment. They offer EI sources for GCMS instruments from various manufacturers. Third one is inlet system. The inlet system connects the ion source to the mass analyzer and allows the ion to enter the system of mass spectrometer. It may include ion optics and ion guides to focus and direct the ions. Fourth one is mass analyzer. We will also discuss previous about this. This component separate ion based on their mass to charge ratio. Different type of mass analyzers are used in mass spectrometry, including magnetic sector analyzer. Ions are deflected by magnetic field based on their mass over charge ratio. Then quadrupole analyzer. Ions pass through a series of 4 parallel rods and the trajectory is determined by the voltage applied to these rods. Then time of flight analyzer. Ions are accelerated to a known energy and then measured based on their time of flight through a drift region. Then a Fourier transfer ion cyclotron resonance analyzer utilizes a magnetic field and radio frequency excitation to measure the mass over charge ratio with higher resolution. Then orbit trap analyzer traps ion in an electrostatic field and measures their mass over charge ratio based on their orbital frequencies. Ion trap analyzers ions are trapped in three-dimensional space using electromagnetic fields, and their mass over charge ratio is determined by the oscillation frequencies. Companies like SCIEX, Thermo Fisher Scientific, and Agilent Technologies offer resources. Then mass separation. The mass analyzer separates ions based on their mass to charge ratio. Mass to charge ratio determination. It measures the mass over charge ratio of ions passing through it, allowing for the determination of their mass. Mass resolution. It provides the ability to distinguish ions with closely spaced mass over charge values, offering high resolution measurements. These are the functions of mass analyzers. Then further, isotopic analyzers. Isotope analysis. Mass analyzers can distinguish isotopes based on their slight differences in mass, allowing for the isotope ratio analysis and studies of isotopic labeling. Then fragmentation analysis. Certain mass analyzers such as tandem mass spectrometers can perform fragmentation analysis, providing insights into the structure and connectivity of ions. Then high resolution measurements. Mass analyzers with high resolution capabilities, allowing for accurate determination of masses even for ions with complex spectra or closely spaced peaks. Fifth one is detector. The detector detects and measures the ion that pass through the mass analyzer. Common detectors include, first of them is electron multiplier. It converts ions into electrons, amplifies the signal, and detects them electronically. Then photomultiplier tube converts ions into photons, amplifies the light signal, and detects them electronically. Hamastow Photonics, Bodley, and Hamline Electronics are examples of companies producing photomultiplier tube. Then microchannel plate detector consists of plate with tiny channels that multiply the number of ions and convert them into electrons for detection. Then ion counting detector registers individual ions, impacts, and count them to determine their abundance. Then charge coupled devices. CCD detectors utilize an array of light sensitive pixels to detect and convert into signals. Mass spectrometry. This is a basic diagram of detector, how they work in mass spectrometry, and detect the mass which is heavier and which is lighter and then falls. The companies which made detectors are, first of them is Hammad Photonics. This is a Japanese company that specialized in optical and photonics technologies. They produce a wide range of detectors including photomultiplier tubes, silicon photomultipliers, photodiodes, photodiode arrays, and image sensors. Then Sony Corporation, we all know about this company, is well-known multinational conglomerate that produces a diverse range of consumer electronics, entertainment products, and components. Photonics is a global manufacturer of electro-optic company and detectors. Burley, previously an independent company, is now a part of L3 Harris Technologies. They have expertise in manufacturing photomultiplier tubes and related products. Then On Semiconductors is a semiconductor solution company that offers a broad portfolio of electronics components and systems. Sixth one is vacuum system. Mass spectrometers operate under high vacuum conditions to minimize interactions between ions and gas molecules. The vacuum system typically includes vacuum pumps, vacuum chambers, and pressure control devices. This is a diagram of vacuum system. 100 electron volt energy is provided and the process occurs. Then seventh one is data system. Data system controls the instruments and manages the acquisition and processing of mass spectral data. It includes electronic software and a computer interface. The software provides tools for data acquisition, processing, analysis, and visualization. Then mass calibration system. Mass spectrometers require calibration to accurately determine the mass to charge ratio. Calibration standards, which are compounds of known mass, are used to calibrate the instruments and establish a mass scale. Ninth one is power supply. Provides a necessary electrical power to operate the mass spectrometer and its components. Includes the ion source, analyzer, detector, and electronics. Then gas supply. Mass spectrometry often require various gases for ionization, collision-induced dissociation, or other processes. Common gases used include nitrogen, helium, argon, and methane. Then control and interface system. These systems enable the user to control and monitor the mass spectrometer operation, including setting parameters, adjusting settings, and interfering with external devices. Then cooling system. Some components of the mass spectrometer, such as the ion source and the mass analyzer, may require cooling to maintain stability and optimal performance. Cooling system can include fans, heat exchangers, and temperature control devices. This is the components of mass spectrometer. First time is sample introduction in inlet, then ionization occurs in ion source, then electromagnetic separation on the basis of magnetic and electric field in vacuum system, then detectors detect them, and signal processes on the computer screen. Additionally, some mass spectrometers may have traditional components or features such as collision cells. These are used in tandem mass spectrometry to perform fragmentation of selected ions, allowing for structural analysis of the molecules. Second one is gas chromatograph. In gas chromatograph mass spectrometry, a gas chromatogram is coupled to the mass spectrometer, allowing for the separation of sample components before ionization and analysis. Then liquid chromatography. In liquid chromatography mass spectrometry, liquid chromatograph is coupled to the mass spectrometer, enabling the separation of sample components before ionization and analysis. Then we see the preparation of these components. First of them, design and development. The designers firstly take the concept of instrument. The first step is to define the purpose and specifications of the instrument. This includes determining the desired mass charge, resolution, sensitivity, and other performance parameters based on the intended application. Then component selection. Each components of the mass spectrometry, such as the ion source, mass analyzer, and detectors, need to be carefully selected based on their desired performance and compatibility with other components. Then system integration. The selected components are integrated into a coherent system, taking into account factors such as vacuum requirements, electrical connections, and control system. Then assembly. Mechanical assembly. The instrument's frame, vacuum chamber, and support structures are assembled. This involves precision machining, alignment, and mounting of components. Electrical connections. Writing and electrical connections are made between various components, including power supplies, detectors, ion source, and mass analyzers. Vacuum system. A high quality vacuum system is crucial for mass spectrometers. This involves the installation of vacuum pumps, valves, and pressure measurement devices to achieve and maintain the required vacuum conditions. Then cooling and heating. Some mass spectrometers require temperature control for specific components such as electronics or detectors. Cooling system, heat exchangers, or temperature controllers may be installed as necessary. Then calibration and testing. Mass calibration. Mass spectrometer required calibration to accurately determine the mass to charge ratio of ions. This is typically done using non-reference standards with non-mass values, and calibration functions are established based on the reference points. Sensitivity and dynamic range. The instrument sensitivity, detection limit, and dynamic range are tested using standard samples of known concentration. This helps establish the instrument performance in terms of detection limits and linear response. Resolution of mass accuracy. The resolution and mass accuracy of the instruments are evaluated using reference samples with well-defined and mass peaks. These tests ensure that the instruments meets the desired resolution and provides accurate mass measurements. Stability and reproducibility. The stability and reproducibility of the instruments are assessed over time to ensure consistent performance. This may involve running repeated measurements of long duration test. Then software and data analysis, in which first of them is instrument control. Software is developed to control the instrument, including parameters such as ionization conditions, scan range, acquisition rates, and other operational parameters. Data acquisition and processing. Software is implemented to acquire, process, and analyze the data generated by the mass spectrometer. This includes peak detection, deconvolution, spectral interpretation, and quantification algorithms. Then validation and quality assurance. Performance validation. The instrument performance is validated by comparing result obtained from known standard or reference material with established methods or published data. This ensures the instrument meet the required specification for the intended application. Then quality assurance, which is very necessary. The entire instrument assembly and testing process undergoes quality assurance procedures to ensure the instrument function reliability, consistently and safely. This may involve adherence to regulatory standards, quality control checks and documentation. Now we see the assembling of these parts. How these parts are assembled. The assembly of mass spectrometer instruments involve carefully integrating various components to create a functional and efficient system. Here's an overview of how the parts are assembled. First of them is frame and vacuum chamber assembly. The frame or chassis of the instrument is constructed providing the structural support of all components. The vacuum chamber, typically made of stainless steel or aluminium, is installed within the frame. This chamber provides a controlled environment with low pressure condition necessary for ionization and ion manipulation. Ports and flags are incorporated into the vacuum chamber for the installation of various components such as ion source, mass analyzers and detectors. Then ionization source assembly. Depending on the chosen ionization technique which we discussed earlier, such as electron impact, electroscopy, etc., the ion source is installed at the appropriate port on the vacuum chamber. The ion source may consist of electrodes, nozzles or other specialized components specific to the chosen ionization methods. Electrical connections are established between the ion source and power supplies or control system. Then mass analyzer assembly. The selected mass analyzers such as magnetic sector, quadrupole, time of flight or ion trap analyzer is positioned within the vacuum chamber. The analyzer is mounted on precision stages or brackets to ensure precise alignment and stability. Electrical connections for powering and controlling the mass analyzer are established. Then detector assembly. The detector, which may be an electron multiplier, photomultiplier tube or semiconductor based detector, is positioned within the vacuum chamber. Similar to the ion source and mass analyzer, the detector is source mounted and aligned for optical performance. Electrical connections are made to provide power to the detector and to transmit signals for data acquisition. Then vacuum system installation. Vacuum pumps such as turbomolecular pumps, ion pumps or rotatory pumps are installed in the appropriate locations on the vacuum chamber. Are associated plumbing valves and pressure measurements devices are installed to regulate and monitor the vacuum conditions within the chamber. Vacuum seals and gaskets are carefully placed and secure to ensure airtight connection between components. Then electrical and control system integration. Wiring and electrical connections are established between the various components, power supplies and control system. Control panels and user interface are installed, allowing operators to set parameters, control instrument functions and monitor performance. Data acquisition system and software are integrated to enable real-time data collection and analysis. Cooling and temperature control. If necessary, cooling systems such as heat exchangers, thermoelectric coolers or liquid cooling circuits are installed to maintain optimal operating temperatures for specific components. Temperature sensors and controllers may be implemented to monitor and regulate the temperature of critical components. Now we see software in mass spectrometry. Software details of mass spectrometry. Mass spectrometer instruments rely on various software components to control instruments, parameters, acquire data, and perform data analysis. Here are the key software details involved in mass spectrometry. First of them is instrument control software. Instrument control interface. This software components provides a user-friendly interface for controlling the instrument's operation parameters. It allows user to set ionization condition, select scan ranges, adjust acquisition rates, and specify other relevant instrumental settings. Then hardware control. The software communicates with the hardware components of the mass spectrometer, including ion source, mass analyzer, detector, vacuum system, and other peripherals. It sends commands and receives data from these compounds to ensure coordinated operation. Then real-time monitoring. Instrument control software often includes real-time monitoring capabilities, allowing users to observe instrument performance, monitor vacuum levels, and track key parameters during data acquisition. Then data acquisition software. Raw data acquisition. This software components facilitates the acquisition of raw mass spectrometry data generated during an analysis. It coordinates the timing of ionization, mass analysis and detection, and captures the resulting data in real time. Data file formats. Mass spectrometer instruments generate data in specific file formats such as MZML, MZXML or proprietary formats. Data acquisition software handles the formatting, organization and storage of acquired data in these standard or instrument specific file formats. Then data processing. Basic pre-processing steps such as baseline correction, noise reduction, and peak picking may be implemented in the data acquisition software to enhance the quality of required data. Then data analysis software, spectral processing. Mass spectrometry data analysis software typically includes function for spectral processing such as deconvolution, smoothing, peak alignment, and calibration. These operations help improve the quality of mass spectra and facilitate subsequent analysis steps. Then identification and annotation. Software tools are available to match acquired mass spectra against databases of known compounds, enabling the identification of molecule present in the sample. These tools often utilize spectral libraries, fragmentation, pattern analysis, or machine learning algorithms for identification purposes. Then quantification. Software facilitates the quantification of identified compounds by comparing their signal intensities to internal or external standards. Various quantification algorithms, including label-free quantification or isotope labeling methods, can be implemented. Statistical analysis. Mass spectrometry data analysis often involves statistical analysis to identify significant differences or correlations between multiple samples or experimental conditions. Tools for hypothesis testing, fold change analysis, principal component analysis, and other statistical methods may be incorporated. Then visualization. Software provides visual representation of mass spectrometry data, allowing users to explore and interpret the results. Spectral plots, chromatograms, heat maps, volcano plots, and other graphical displays aid in data interpretation and presentation. Data management and integration. Data storage. Software system may include capabilities for secure and organized storage of mass spectrometry data. It allows user to store, retrieve, and manage large volume of acquired data efficiently. Then data integration. Mass spectrometry data is often integrated with other omics data or metadata to provide a comprehensive understanding of biological system. Software tools enable integration with genomics, transcriptomics, proteomics, or metabolomics and data facilitating multimix analysis. Then vendor specific software. Mass spectrometry manufacturers often provide proprietary software suites tailored to their instrumental modes. These software packages include instrument control, data acquisition, and data analysis capabilities specific to their instruments. They may offer advanced features and customization options designed to maximize instrument performance. Now we see the applications and benefits. First of them is compound identification in complex mixture. Compound identification in complex mixture is one of the primary applications of mass spectrometry. Complex mixture often contain a wide range of compounds, making it challenging to identify and analyze individual components. Mass spectrometry offers several advantages in their regard. First of them is mass to charge ratio. By comparing the measured mass over charge values with reference databases such as spectral libraries, the identity of the compounds can be determined. Then fragmentation patterns. Fragmentation patterns provide additional structural information about the compounds, adding in their identification. By comparing the observed fragmentation patterns with known patterns, researchers can reduce the chemical structure and functional groups present in the compounds. High sensitivity. Mass spectrometer is highly sensitive, capable of detecting compounds even at very low concentration. The ability to detect and quantify compounds at low levels makes mass spectrometry a valuable tool in various fields, including environmental monitoring, pharmaceutical analysis, and clinical diagnostics. Third, selectivity and specificity. Mass spectrometry offers high selectivity and specificity in compound identification. The combination of mass analyzer and detection methods allows for precise separation and measurements of ion based on their mass over charge values. This selectivity ensures that only the target ions of interest are detected, minimizing interference from other compounds present in the mixture. Quantitative analysis. Mass spectrometry enables quantitative analysis of compounds in complex mixture. By measuring the abundance of ions, researchers can determine the concentration of specific compounds. This quantitative capability is important in fields such as pharmaceutical research, where accurate measurement of drug concentration is essential for determining efficiency and safety. This is the diagram of chromatogram for specific ligand, in which in this ligand receptor complex formation, then ligand detection, then ligand dissociation, and then ligand receptor complex separation, and at the end we see the data in the form of graph, ligand binding curve. Quantitative analysis with high sensitivity and accuracy. Mass spectrometry is widely used for quantitative analysis due to its high sensitivity and accuracy. Here's an explanation of how mass spectrometry achieves quantitative analysis. Calibration standard. Quantitative analysis using mass spectrometry typically involves the use of calibration standards. These are known concentration of target compounds that are analyzed alongside the sample. By measuring the response of the calibration standards, a calibration curve can be generated which relates the signal intensity or ion abundance to the concentration of the compound. Then isotope dilution. Isotope dilution is a powerful technique used in mass spectrometry for accurate and precise quantitative analysis. It involves adding a non-compound amount of isotopically labeled internal standard to the sample. Before analysis, the internal standard has a similar chemical structure to the analyte but differs in its isotopic composition. Then selected reaction monitoring and multiple reaction monitoring. SRM and MRM are targeted mass spectrometer techniques used for quantification. Specific precursors ions and their corresponding product ions selected to detection provide high sensitivity and sensitivity. By optimizing the instrument parameters to monitor the specific precursors to product ion transition of the targeted compounds, accurate quantification can be achieved even in complex matrices. Third is structural elucidation of molecules. Mass spectrometry is a powerful tool for structural elucidation of molecules. It provides valuable information about the composition, connectivity, and functional group present in a compound. Here's how mass spectrometry contributes to structural elucidation. First point is mass to charge ratio. Mass spectrometry measures the mass to charge ratio we discussed earlier of the ion generated from the sample. The mass over charge value provides important clues about the molecular formula of the compound. By comparing the measured mass over charge value with the calculated mass over charged values of possible molecular formulas, the molecular formula can be determined. Fragmentation patterns. In techniques such as tandem mass spectrometry, the ions are further fragmented, producing characteristic fragmentation patterns. The fragmentation patterns provide valuable information about the chemical structure and connectivity of the compound. By analyzing the fragmentation patterns and comparing them with database of non-fragmentation pathways, the structure of the compound can be deduced. Then ion activation methods. Different ion activation methods such as collision-induced dissociations, electron capture dissociations, and electron transfer dissociations can be employed in mass spectrometry to induce controlled fragmentation of ions. These techniques generate specific type of fragment ions, aiding in the determination of structural features like functional groups, bond cleavages, and stereochemistry. The following diagram displays the mass spectra of three simple gaseous compounds: carbon dioxide, propane, and cyclopropane. Now we see about the costing of mass spectrometry components. Entry-level instruments. Entry-level mass spectrometers such as benchtop models or compact system typically range from $100 to $300. These instruments may offer basic functionality and lower performance specification compared to high-end models. Then high performance instruments. Spectrometers which offer advanced features, higher resolution, increased sensitivity, and expanded capabilities can range from $300,000 to several $1 million. These instruments are often used in research laboratories or specialized applications. Then hybrid instruments. Hybrid mass spectrometers such as quadrupole time of flight or D-trap instruments which combine multiple mass analyzers for enhanced performance and versatility generally fall in the higher price range. Prices for hybrid instruments can range from $5 lakh to several $1 million. Then specialized instruments. Mass spectrometers designed for specified applications such as proteomics, metabolomics, or imaging, which have specialized features and higher cost. Prices for these instruments can vary widely, ranging from hundreds of thousands to millions of dollars depending on the specific requirements. Thank you so much.